Hello there, welcome to this module in which we will look at the cell structure. You can consider this chapter as a little bit of an introduction where we will review the key concepts regarding cells in general and then look at different parts of the cells and cell organelles. Early on, we will also make a reference to the methods of visually studying the cells, microscopy. So we are going to start by jumping into this listing of the levels of organization. You can see here subatomic particles, electrons, protons, and neutrons. An atom, molecule, mitochondria, and a cell, followed by tissue, which were made of many cells, an organ, and organ system. And of course, an organism, such as you, as an independent living creature, is made of multiple organ systems. So let's have a look of our cell level a little bit more. That's what we will be focusing on today. Here we have been defined these as fundamental basic units of life. What does that really mean? Well, they are the smallest individual unit that can function independently at the smallest level. And you will see that the cells are small, typically about 1 millimeter at the largest, and smallest about 1 micrometer. Remember, we learned about the metric scale in our previous lab. What's to note here is that actually the scale may differ from that, but that is the general scale that most cells fall into. A few odd examples include, for example, nerve cell in our spinal cord, which can be at the length even up to 1 meter. But this is more of an exception than a rule. What I find fascinating is that really how we start as single cells, and then as the cell division progresses, cells specialize and become cells of different types. For our nails, hair, photoreceptors in an eye, cardiac cells in the heart, nerve cells in the brain, and so on. It is really quite a remarkable journey. But it all is all about these individual cells. They are the smallest scale of life that can function independently. And there are many different kinds of cells. Here you can see as an example cells from the nasal sinus, onion cells, and finally bacterial cells. They are all from very different organisms, but yet they share many similar characteristics, such as the cell structure. In fact, we have been studying cells since 1830s, when most of this work was conducted with a naked eye. This limited the cell study mainly to very gross characteristics such as shape and maybe seeing a nucleus too. As technology developed, we became able to do some more detailed study. Can you guess what was this breakthrough thanks to? Well, if you said microscopes, you are correct. So we have gotten far from the early microscopes, but the basic concept remains the same, especially when it comes to the light microscope. So as you can see, the scale of light microscopes is larger, somewhere around nanometers to millimeters. While the scale for the electron microscopy, where instead of using light beam that travels in the microscope, we use electron beam, which allows us to go to a much smaller scale. There we are talking about a scale of 0.1 nanometers to the overlapping with that of a light microscope. I think it's always a good thing to put things into scale. So what we see here is where cells would fall on this scale. Other thing that you will notice is that there is a fair bit of an overlap between these methods of looking at things. Between what we can see with a naked eye and light microscope, and then again between light and electron microscope. But really, to me, it is quite amazing that we are now just at the break of being able to start to study single atoms with the best electron microscopes. I was just in a web conference where we were shown first images of an individual atom as produced by an electron microscope. It was quite impressive, so I am excited to see what the future holds in this field. So, as we already concluded, cells are typically always rather small. Let's take a moment to consider why is this the case. And this has all to do with a concept of surface area to volume ratio. So what we see here is that when the size of the cell grows, the relative amount of the surface area compared to its volume decreases. Why does this matter? Well, a lot of things that the cell does take place on the cell membranes. So having as much cell surface area is important because that surface area is all that the cell has to deal with the contents that made up the rest of its volume. If we look at the small cells here on this illustration, they have more surface area, while the larger cell has less when compared proportionally to the volume of the cell. Another way to think about this is to think about a fuel truck as an example. So if the cell grows too big, traveling distances from the cell membrane to the inner parts of the cell grows too long, just like the truck, which transports fuel and needs some fuel itself to drive the distance. Similar is same for the cell organelles. If the distance grows too long, they end up using the resources that they are supposed to be simply transporting. I hope that that makes sense. Some of my students prefer to think about this concept in this way. So the final thing for us to consider is that there are any other ways how cell could get more surface area. After all, we know that having lots of surface area was ideal for us. Right. Well, we can make folds to the cell membrane. By increasing the surface area, we are now able to conduct more of the business that was conducted on the cell's membrane only. So that would be beneficial, right? So I guess that the last question that I have is that what can the cells do when they reach too large of a size? Other than making these folds that we just discussed about. Well, we find out that when there is insufficient surface area to support a cell's increasing volume, a cell will either divide into two or die. So that is what cell can do too. Well, there is another few terms that I want to make sure that we feel good about. So when we are talking about study of cells, we talk about cytology. So studying individual cells. And when we study entire groups of tissues, we talk about histology. Please make sure that you are clear of this. And in case you were wondering what we are seeing here, for the individual cells, we see normal, healthy uterine cervix cell from a pap smear, and from the tissues, Salmonella bacteria, which shows up as these tiny purple dots. But that is not something that I would expect you to know. I just think that it is nice to know what you are looking at. So when we look at a cell, we see all these small structures inside it. These are known as cell organelles. And sometimes students ask me a question, how did the first cell come about? Or how did it end up having these things inside it? Well, that is a great question. And there is still some uncertainty about this, but I am going to give our very best go in at least partially trying to address this. First cells that we had were probably really simple kind of cells, like primitive prokaryotic cells. And you will notice that when we talk about these prokaryotic cells and their characteristics more later on, so for now, just focus on the story of cells. We think that they were probably not much more than just some organic compounds like simple RNA strands surrounded by a membrane. And over time, more compounds developed like DNA and proteins, and the cells started to evolve more complex structures. Remember, these were still very simple considering what we think of a typical cell these days. Cells became more stable and able to reproduce itself and pass genetic information to the next generation. This was life starting to appear. Remember our characteristics of being something that is living. And this figure here shows our current understanding of what the earliest cell may have looked like. We think that by having this membrane around the molecule, these molecules were prevented from reacting with other molecules as would have normally happened. So these molecules persisted instead. And that was really the start of the first cells. There is no doubt that there were many and many different kinds of early cells of this kind. However, scientists tend to share a view that only one early cell or a group of cells were responsible for eventually giving a rise to all subsequent life on the Earth. And we call that cell the last universal common ancestor, LUCA. And you might have seen these trees of life where we show the connections between different species and their evolution. Well, LUCA would be the very first point on these trees. And the exciting part is that we do not yet know what that point was like. So that is a very inspiring topic to research. So what happened then? We think that the earliest cells got their energy from other molecules that were floating in their surroundings. However, about 3 billion years ago, a new way of getting energy appeared: photosynthesis. And we will talk more about photosynthesis later in this course, but to put this story short, these cells were able to do the photosynthesis and could use sunlight to make food from carbon dioxide and water. This was huge because they provided food for themselves. And this caused a bit of a disaster as oxygen started to build up in the atmosphere, and this oxygen was at the time poisonous to most early cells. After all, they had evolved in its absence, so they died out and only very few survived. And then a second major innovation took place: cellular respiration came about. And again, we will talk more about it later in this course. But with this, these cells were able to use oxygen to gain more energy from organic molecules. But I think that we have still not really answered how all these organelles came about inside the cell. After all, based on what we have talked so far, it would have looked more like this. Well, what we think is that something that is now known as an organelle accidentally ended up inside the cell. And it turned out that by it being inside the cell, both the cell and this what was to become an organelle both benefited from it. Or at least one benefited and another one was not really bothered. And as they were in this symbiotic relationship, that was useful and over time became more common. And this way, more and more organelles came about. And then we have our cell as we now know it. Well, that was a fun story, but not one that I would expect you to know in detail, just simply the big picture of organelles appearing initially as a result of a symbiotic relationship. So, before we wrap up this video, I want to speak about one more concept. So we can group cells in two main types, something known as prokaryotes or as eukaryotes. And we saw this prokaryote already earlier. Just quickly, let's start with that. So prokaryotic cells were the first to evolve. They first appeared way before this other cell type, so they have been around much longer. Another way how I hear them being described is that they are less evolved. And that is true, they are fairly simple. But that does not mean that they would be bad or less impressive than more complex cells. In fact, these prokaryotic cells have been around for a long time and are still very successful all around us. So they must be doing something right. And their small size and relative simplicity might be the very reason for this. This allows them to reproduce very quickly and reach large numbers very fast. There are some that are harmful, such as this norovirus bacteria that I have shown here, while others, such as this baker's yeast that I have shown here, can be useful. In fact, it is the case that most of these are beneficial or harmless for us, and only a very small portion is harmful. So I would go as far as saying that prokaryotes have been and continue to be very successful on their own right. One thing that I do want to highlight regarding prokaryotes is that they do not have membrane-enclosed nucleus. Instead, their genetic material is free in the cytoplasm. And then, if we look at the bacteria, they can have a protective cell wall or a layer, which often gives them the shape of the cell. And again, we see this same characteristic. No membrane-closed nucleus. But we will see ribosomes, which serve the role of the protein synthesis, and we will talk more about those soon. And one thing that we often see with these is that they can have structures coming out of the cell wall for movement, attachment, or for example, passing DNA to another cell. And let's move on to have a look at the eukaryotes. So these are the cells that we often think of when we think of the cells. They are significantly larger in size. In fact, this comparison here is not probably quite to scale. In addition to what we see here is that they have a lot of specific compartments inside them. So different functions and different cell organelles are housed at different parts of the cell. And one example of these membrane-enclosed compartments is our nucleus. So the DNA is stored in a eukaryotic cell inside a nucleus. This is a membrane-enclosed structure, much different from the prokaryotes. And these organelles that we see numerous in a eukaryotic cell, they serve different purposes. It can be, as we saw in the case of a nucleus, storage of genetic material, or a membrane system, and we will talk more about that in a bit too. Or these organelles can be all about energy production and use, or they can be there to simply provide structure. So a lot going on here. I hope that this comparison of the two makes the difference between them a little clearer. And this is actually where we are going to wrap up this video, and we will continue in the next video by reviewing different cell organelles. I will see you then.